In this lecture, we're going to talk about cells. The first thing we're going to talk about is, where do cells come from? Have you ever thought about that? We know that cells exist, but how do we prove where they come from? Well, a couple hundred years ago, it wasn't so easy. A lot of people believed that cells came from spontaneous generation, and what that means is that cells just arise out of nowhere. Like they could arise from even non-living things. And then in the mid 1800s started to come the belief of cell theory, where cells arise from pre-existing cells. Now we know that that's true, but it took scientists of that day a long time to convince people of that. One of the more famous experiments in disproving spontaneous generation and proving cell theory was Francisco Redi's meat experiment. There were a few others as well. Essentially what he did is he took some meat and left it in a container, and he left that container either open to the air, closed from the air, or with a screen on top, or a mesh gauze, if you will. So in the container that was open to the air grew maggots and flies. In the container that was sealed off from the air, nothing happened. However, critics said that maybe that's because there's no air in that container. So then he did the experiment again with the container with the gauze on top, and nothing grew on the meat, no maggots, no flies. And so what that helped prove was that these maggots and flies weren't just arising out of the meat randomly, that they existed somewhere else and came there. So that helped steer scientists away from spontaneous generation. So we know today that cells come from pre-existing cells. So there are two overall types of cells, you guys know that, prokaryotes and eukaryotes. Prokaryotes make up bacteria and archaea. Eukaryotes make up all of the protists, fungi, plants, and animals. Right, so I'm hoping that by the time you're taking this course, you've already learned a bit about eukaryotic cells and you're kind of familiar with them through either A&P or other science classes you might have taken, even all the way back to high school. So what I'm really going to focus on are the features that are unique to prokaryotes and the differences between prokaryotes and eukaryotes. But towards the end of this lecture, I will go over some of the eukaryotic organelles and those sorts of things. All right, so whether you are a prokaryote or a eukaryote, you have a plasma membrane or a cell membrane, same thing, very frequently called both of those things. DNA, your genome, right? You have cytoplasm, and you have ribosomes, okay? Some characteristics of prokaryotic cells specifically: they're much smaller than eukaryotes, right? They have a genome made of DNA, but it's not inside a nucleus, and that's one of the hallmarks of a prokaryotic cell, right? Eukaryotes have a nucleus bound by a nuclear envelope. Prokaryotes don't. They just have their DNA kind of bunched up in the middle of their cell. That is actually a region called the nucleoid region, meaning it's kind of like a nucleus, but not. And in that nucleoid region, the DNA is condensed, but it doesn't have a membrane around it. What prokaryotic DNA really is it's one big circle of DNA. You're probably familiar with the human genome, where you have lots of little lines of DNA. Those are called linear fragments of DNA. Prokaryotes don't have that. They have one big circle that they kind of bunch together in a ball and put into the nucleoid region. Now, the nucleoid region could be anywhere inside the cell. We're just referring to kind of where the DNA is. Another unique thing about prokaryotic cells is that they divide by this process called binary fission. And what binary fission is a duplication of the DNA, and then the cell just splits into two. There's no mitosis, no meiosis. There is no choreographed process with checkpoints and other things like that. It's not fancy at all. It's just duplicate the DNA and split into two. There's a great video showing you an animation of that I'm going to put into our module on Canvas. The next thing we're going to talk about are cell morphologies. And what a morphology is, it's a shape or an arrangement of cells. And with bacteria, there are a few different ones. And that's actually pretty cool. The first one we're going to talk about are circular cells. We call those foci or coccus cells, meaning that they're round or circular. They're spherical. They're not always a perfect sphere, but they're spherical. So some more famous bacteria that are foci shaped are Staphylococcus aureus. That is a bacteria that lives naturally on human skin. It has developed some antibiotic resistance and turned into what we call MRSA, or methicillin resistant Staph aureus. It's also developed resistance to a few other drugs as well. We have Streptococcus pyogenes. Now these cells, it's kind of hard to see, but these are chains of little circular cells. Strep pyogenes is a very famous human pathogen known for causing infection in multiple places in the body. Strep throat, for example, is usually Streptococcus pyogenes. It can also infect many other areas of the body, including the blood, and be a really bad pathogen. Some others, Streptococcus pneumoniae. These are either single circular cells or short chains. They are one of the many bacteria that can cause pneumonia, hence the name, but can also cause other infections as well. Also in our foci-shaped cells, we have a couple of Neisseria genus of bacteria. We have Neisseria gonorrhea and Neisseria meningitidis, which is kind of like, in the same sense as pneumonia, one of the many bacteria that can cause meningitis. Our next cell shape are the rod-shaped bacteria. They're kind of like long ovals, cylinders with kind of rounded edges. We call those either bacillus cells or bacilli if they're more than one. You're going to find a lot of bacteria are bacillus shaped. Now there's also a genus of bacteria called bacillus bacteria, and they're all bacilli shaped, but those are not the only ones. Some pretty common bacteria that are bacillus shaped are Pseudomonas aeruginosa. You can see this is two rods together, and these guys actually have these big long flagellas at the end that you can see in this image. Salmonella. There are lots of kinds of salmonella, actually, not just Salmonella typhi that causes the food poisoning that we're all familiar with from chicken and things. But Salmonella is a bacillus shaped bacteria. E. coli, also bacillus shaped. Y. pestis, Yersinia pestis. This is the causative agent for the Black Plague. That is a bacillus-shaped bacteria. And Listeria monocytogenes. This is a really great picture. This is a really long, rod-shaped bacteria. And you can actually see some of the features around the outside and inside the cell in this image as well. Listeria is often a food contaminant, especially in deli meat and produce. It can actually also be bad for women who are pregnant, which is why they tell you not to eat deli meat when you're pregnant. Some other less common shapes of bacteria, we've got Vibrio and Coccobacillus. These are kind of variants of rod-shaped bacteria. Vibrio are curved rods, so they're kind of rod-shaped, but they've got a pronounced curve to them. And then we have Coccobacillus, which are kind of a cross between a rod and a foci, but it's not spherical enough to be considered a foci. Our Vibrio, one of the more famous Vibrios are Vibrio cholera, causative agent for cholera, and a Coccobacillus Bordetella pertussis. So this bacteria is the one that causes whooping cough. You can see it's not really a true bacillus, but it's not a true foci either. So we call that Coccobacillus because it's in the middle. Our final category are the spiral-shaped bacteria. There are two kinds, Spirillum and Spirochete. Spirillum have a rigid body, and Spirochetes have a flexible body. The Spirillum, in order to move throughout their environment, they have flagella at one or both ends of the long spiral. They usually live in water, often water with organic matter present. Okay? And then our Spirochetes are similarly shaped, but they have a flexible body. And they actually have what's called an axial filament to move, which is kind of like a specialized flagella that wraps around the outside of the cell and causes it to move like a corkscrew. Here's an example of a Spirillum bacteria. It is Spirillum pleomorphum, Spirillum voltans. It's just a couple of the Spirillum genus of bacteria. These bacteria are microaerophiles, meaning that they need oxygen, but only in small amounts, and we'll learn more about that later. They're found usually in polluted pond water, and they have these tufts of flagella at the end of their rigid bodies. There's a great video here that I will put in our module about how Spirillum move. They kind of use their flagella to swim through the environment, okay? And one thing I wanted to point out is you can find facts about these microbes, including maybe a microbe you want to use for your term paper, at Microbe Wiki, which yes, it's a wiki, but it's a wiki run by a college, and it is very credible and has great information about microbes in it. And I put the link down there for you as well. And this picture is really cool because you can really see those tufts of flagella, or a lot of flagella in one place at one time. Now, Spirochetes are a little bit different than Spirillum because remember, they have flexible bodies and they have this axial filament, which is a big long flagella that comes out of one end and wraps itself around the bacteria. Here's a better picture. Okay, so the axial filament wraps around, and when the flagella or the axial filament moves, it causes the bacteria to kind of spin like a corkscrew, and that's how it moves through its environment. A couple of more famous bacteria in this category are Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease. And I have, again, a short video clip of how these bacteria move. And you can see when you watch this, just how different the movement is between a Spirillum and a Spirochete. The next thing we have to talk about when we talk about cell morphology is how they're arranged, because different species of bacteria arrange their cells in different formats. And that can be one thing that's useful in identifying bacteria. We're going to talk about some prefixes that we can add to the end of, to the beginning of the word of the bacteria. So coccus, shape or circular shaped, right? You can have just plain old cocci, or you can have streptococcus or staphylococcus. Streptococcus are chains of cocci. Strepto means chains. Or you can have staphylococcus, which are clusters. Okay, so these cells aren't really on their own, they grow in kind of clumps. So strepto, chains, staphylo, kind of clusters or clumps, if you will. You can also have two cocci together, and they commonly only exist as one or two together. Those are called diplococcus, di for two. Sometimes you'll see four, that's a tetrad. Those are more rare, but most commonly you're going to see single chains or clusters. Now, Bacillus won't do any of that clustering or anything. It can be by itself, or it can make chains. And a chain of Bacillus cells would again have that strepto prefix and be called Streptobacillus. All right, let's talk about the parts of prokaryotic cells. I'm going to cover all of the parts of prokaryotic cells, even the ones that eukaryotic cells also have. Okay, something unique to prokaryotes is their cell wall structure. Now, we know that cell walls are located outside of the plasma membrane, right? They're kind of the most external part of the cell. They're usually a very, very strong structure. They help protect the cell. They help maintain its shape, and they protect it from osmosis or osmotic damage in environments that either have too much or too little salt. And now those environments are called hypertonic or hypotonic, and you might have heard of them before, especially in A&P. So a little bit about that in case you haven't heard of it before, or it's always a good thing to review. The cell wall is important because it helps protect against osmotic lysis. I can't stress that enough. So osmotic lysis happens when water moves either into or out of the cell because the concentrations of solute, or salt most commonly, are greater either inside the cell or outside the cell. If the salt balance is not equal, water moves to try to balance the solute concentrations. The water moves, the salt does not move, okay? If there's more solute outside the cell, water moves out of the cell. This causes the cytoplasm to shrink, which is called plasmolysis. This kind of solution is called a hypertonic solution. Hyper, there's lots of it. There's lots of salt outside the cell, or solute outside the cell, okay? The opposite of that, if there's more solute inside the cell, meaning the environment outside the cell is very diluted or very much not salty, water moves into the cell to try to balance out the salt concentration inside the cell versus outside. This causes the cell to swell, blow up like a balloon, usually to the point of bursting or lysis. These solutions are called hypotonic solutions. Hypo, there's less salt outside. The biggest thing to remember is that water follows salt, or solute, what have you. Where the salt is higher, the water goes. If the salt is higher outside in a hypertonic solution, water goes out. If the salt is higher inside in a hypotonic solution, water goes in, okay? What a cell wall does for us is it helps protect against the flow of water. It slows it down. It doesn't stop it completely, but it supports the plasma membrane, so it's less likely to burst, right? That can really help a cell out. Here's a picture of the same things. Great study guide for you here, showing you which way the water goes, okay? So bacterial cell walls are really unique. They are made by this substance called peptidoglycan, which is a very, very important substance and very unique to bacterial cells. Only bacteria have peptidoglycan in their cell walls, and all bacteria have peptidoglycan. So it's one of those hallmark features of bacteria. It is a very strong structure made of sugars and amino acids kind of bound together in this big mesh framework. So we say that peptidoglycan has a disaccharide polymer backbone. A disaccharide are the complex made of two different simple sugars put together. Those simple sugars for peptidoglycan are called NAG and NAM. And a polymer means that those two simple sugars together are repeated over and over and over again throughout the length of the molecule. Those polymers of sugars, which are these long cylinders here, are then linked together for stability and to provide that kind of mesh framework by little short amino acid chains. And the short amino acid chains that are too small to be proteins are called peptides. And there are 4 specifically in each one of these little green chains. So we call that a tetrapeptide, 4 amino acids. And what that does is it links all of that, the NAG and NAM together, and provides that strong meshwork holding all of the peptidoglycan together. Something that's really interesting is that some antibiotics can actually interfere with these cross bridges to weaken the cell walls of bacteria. Now there's one more feature of some bacterial cell wall peptidoglycan, okay? And that exists in gram-positive cell walls. And that feature is called teichoic acid. And that's these kind of little black squiggly lines that come up through the peptidoglycan. So if this is our plasma membrane of a bacterial cell, outside it you have many, many layers of peptidoglycan. And then in gram-positive cell walls, meaning that for a special kind of stain called a gram stain, they will always stain positive. They have lots of peptidoglycan and then this teichoic acid coming up through, and that kind of anchors it to the cell's plasma membrane. Now, gram-positive cell walls are therefore very, very strong. Okay. Teichoic acids, however, are susceptible specifically to penicillin. So a lot of times gram-positive bacterial infections will be treated with penicillin or a derivative of penicillin. The bacterial cell walls can either be gram-positive or gram-negative. It's one or the other. Gram-negative cell walls look a little bit different than gram-positives. Picture gram-positive, right? Lots of peptidoglycan, many, many layers. Okay. Gram-negative cell walls have a very, very thin layer of peptidoglycan, and then on the outside of that peptidoglycan, they have another plasma membrane. And this plasma membrane is called the outer membrane. It is a phospholipid bilayer, just like the cell's plasma membrane, except that this also has some very important proteins in it. Porins to let things through, but most importantly, lipopolysaccharide. Lipopolysaccharide is a molecule that can signal to a host immune system that there's an infection or that there's a foreign bacteria in the body. Lipopolysaccharide itself, when it's attached to this outer membrane, is not really harmful to the host. It's not also very well recognized by the host, but when these bacteria start to die inside their host, the little bits of the cells start breaking up and floating around inside the host's environment. And when the lipopolysaccharide breaks up and starts floating by itself into the host environment, that is a big signal to the immune system that there's an infection, and it really ramps up the immune response. Now, the space between these two membranes is referred to as the periplasm, right? There's still that kind of cytoplasmy stuff in there, although not to the extent as inside the cell, but there is fluid there, right? It's not just empty. And then again, we have porins or protein channels inside the membrane to let things through. Okay, there's a little bit more about LPS. It has three components: the lipid A part that's actually in the membrane, the core polysaccharide that kind of sticks out like a tail, and then the O polysaccharide that's kind of like a little flag on the end that says, here I am, I'm LPS. Okay, LPS is also referred to as endotoxin. It only exists in gram-negative cells. It can be sensed by the host immune system, like I said, and occasionally it actually can overactivate the immune system, leading to hemorrhaging and septic shock. Here's a great picture of both of those kinds of cell wall types together, either gram-positive or gram-negative. Gram-positive has thick peptidoglycan and teichoic acids. Gram-negative has a thin layer of peptidoglycan and then an outer membrane with lipopolysaccharide or LPS inside it. Okay, and these will be important to know, and we're going to keep referring back to this over the course of the semester. Where does gram come from? Well, it comes from the gram stain, originally made by Hans Christian Gram. He was a Danish bacteriologist, and he was looking for a way to visualize bacteria well under the microscope. And he had no idea what the two different colors meant until many, many years after his death. Okay. There are a couple of other kinds of cell walls that exist in bacteria. There's exceptions to every rule in biology, right? So one of those cell walls are called acid-fast cell walls. They have, again, peptidoglycan, a small layer of peptidoglycan, and then they have a couple of other layers, and their outermost layer is actually really kind of impermeable to anything. It's this kind of waxy layer of a substance we call mycolic acid. I mean, it does not let dye through. So this would be an exception to gram staining in that it would not take up the stain. So you can actually stain it in other fashions by what's called an acid-fast stain. Things of this nature are some pretty pathogenic bacteria, things like Mycobacterium tuberculosis, Mycobacterium leprae, which causes leprosy, which is shown here. So we don't ever get to work with acid-fast cells in a college microbiology lab because they're so pathogenic. Another one are some bacteria, really just the mycoplasmas, don't really have cell walls because they're so tiny. Okay, so this is a picture. These guys are much smaller than normal bacteria, even though you can't really tell that from this picture. And then we have our archaea, which are also prokaryotes, but their cell walls are either completely absent or really unusual. They do not have peptidoglycan. They're not bacteria. They have polysaccharides, glycoproteins, and sometimes this compound called pseudomurin. They will appear gram-negative because everything takes up the safranin stain for gram-negative staining, as you'll see, but they do not have peptidoglycan. All right, damage to the cell wall. So as you can imagine, something that protects the cell could be a target for things that help destroy the cell, right? The cell wall itself or how the cell wall is made are targets for different antimicrobial drugs like antibiotics and other things. Interestingly enough, it's also a target for lysozyme. You might have heard of lysozyme before. It's a digestive enzyme made by most eukaryotic cells, but it exists in things like our spit, our tears, our mucus, our sweat. It's one of the things that our body has that tries to keep invaders out of the body. It actually works by breaking down the peptidoglycan backbone. And by breaking down that peptidoglycan backbone, it makes the cells more susceptible to osmotic lysis. Outside of the cell wall is often the substance called glycolax. It's a really sugary or carbohydrate-rich substance that is kind of slimy. It can be called a slime layer if it is very loosely aggregated. But if it's highly ordered and packed together really tight, it actually makes a capsule around the bacteria, which is kind of a coating of glycolax around the entire cell. Glycolax can help our cells adhere to different surfaces, and it can help them form biofilms. And what biofilms are, we'll talk more about, but they're colonies of microbes that grow on surfaces, and they secrete a lot of glycolax around the outside of themselves to kind of isolate themselves from their environment. And glycolax can also help increase pathogenicity of a microbe because it makes it harder for cells in the host immune system to recognize and take up the bacterial cells. There's also a layer of structural proteins called the S layer. We're not really going to talk about them. They're not well understood. Sometimes they function as cell walls in archaea, but bacteria can have them as well. All right, a couple of more things, appendages outside the cell wall, flagella, which we'll talk about in a minute, but also fimbriae and pili. Fimbriae are kind of short, bristle-like projections. Think of like a burdock or like almost like a Velcro itself to the surface. There can be hundreds of them outside a cell. They're really important for attachment to surfaces. Or you can have pili. Pili are these kind of appendages that cells can actually transmit little bits of DNA through from one cell to another. But the one you're most familiar with are probably flagella. Flagella are long, kind of tail-like appendages. They're used like propellers to move in aquatic environments. They have stiff filaments. They have three parts: a basal body or motor that's inside the plasma membrane. They have a hook that kind of connects the basal body to the filament, and then they have the big long filament or the tail outside of the cell. So basal body, hook, and filament. Something unique about prokaryotes, because eukaryotes can also have flagella, are that in prokaryotes, the flagellas spin like a circle, but in eukaryotes, they kind of flap back and forth in a wave-like motion. There can be lots of different arrangements of flagella. These are just some terms you might hear. Monotrichous means one, amphi means both ends, lithotrichous, or a tuft of flagella at one end, or many at one end, and then peritrichous are kind of all over. All right, moving inside the cell, we have the plasma membrane. The plasma membranes are relatively similar between prokaryotes and eukaryotes. They are a phospholipid bilayer. So it's a phospholipid, which means it's a fatty acid that has two tails and a big phosphorus group on the end. And the fatty acid tails are hydrophobic. They don't like water, which is why they associate inside of the membrane, if you will, and the phospho part are on the outside, okay? Because the phosphorus groups like the water, and the fatty acid tails don't. Now, all of these phospholipids together makes that kind of barrier of the plasma membrane, right? The plasma membrane is what we call selectively permeable. Some things can diffuse through the membrane, but not many. And then there are proteins and channels and pores and lots of other things that control what goes in and out of the cell. What's interesting is we normally think of the plasma membrane as kind of this static or not moving thing, but really within the membrane, things are constantly moving all of the time. I mean, they're keeping their position across the membrane, but they're moving within that layer, and that's called the fluid mosaic model. And I actually have a video clip down here for you of that. And there's lots of things in the plasma membranes of our cells. Proteins, carbohydrates, lots of things. So proteins on the cell's surface are important for cell-to-cell communication. They're important for sensing things in the environment. They can even be things that help make a pathogenic cell more pathogenic, called virulence factors. A lot of them will have sugars associated. So if you have a sugar and a protein kind of put together in one big molecule, that's a glycoprotein. Some of them have sugars associated with lipids in the phospholipid bilayer. Those are called lipoproteins, or proteins associated with the phospholipid bilayer. Many of them extend from the surface of the cell, and they allow the cell to interact with its environment. One difference between prokaryotic cell membranes and eukaryotic are that in eukaryotes we have sterols like cholesterol in our plasma membrane. Those don't exist in prokaryotes. Now, to get through the membrane, we're not really going to focus on this because I know you cover this a lot in A&P. There are ways to be transported across the membrane. Transport can be active or passive. If it's active, it needs ATP. If it's passive, it doesn't, right? Molecules will naturally move from areas of high concentration to areas of low concentration. And they can do this passively through pores, if they're small and uncharged molecules. But if they're large or charged, or they're moving across their concentration gradient, they're going to need some sort of protein powered by ATP to get them across the membrane. We're not going to cover it any more than that. Like I said, I know that you're going to cover that a lot in A&P if you haven't already. All right, let's go inside the prokaryotic cell. First thing we have is cytoplasm, right? Cytoplasm is this mostly water liquid mixture that is inside a cell. But there are so many things inside the cytoplasm, so many proteins and sugars and organelles and other things like that, that there's so much stuff in it, it's almost like a gel inside the cell. It's mostly transparent, so when we look at cells through the microscope, they appear clear in the middle. But there's really a lot of stuff in there, including even some cytoskeleton elements like microfilaments, microtubules. Those are more prevalent in eukaryotes. And then inside the cell, where the genome is, we call that the nucleoid region, right? It's technically an area of condensed DNA inside prokaryotic cells, right? So it can exist really anywhere in the cell. It is anchored to the plasma membrane at one point, so it's not just free floating, but at that point can vary. But it's all in the nucleoid region. Some prokaryotes can also have little extra pieces of DNA called plasmids. So the main chromosome is a very large circle of DNA. It never looks like this inside a cell. It's always clumped together. But it can also have these smaller little rings of DNA called plasmids. And plasmids can carry very unique genes on them for like toxins or antibiotic resistance or things like that. And plasmids can be transferred from one cell to another or replicated into another cell. So they are a unique structure of DNA that can exist in bacteria. Prokaryotes have ribosomes, just like eukaryotes do. Ribosomes are where proteins are made. There are two ribosomal subunits, the small subunit and the large subunit. They kind of make a sandwich around the mRNA to make the proteins, right? A difference between prokaryotic and eukaryotic are that the ribosomes are slightly smaller in prokaryotes. We give the subunits little names based on their size. So 50S for the large subunit and 30S for the small subunit. In prokaryotes, in eukaryotes, it's 60 and 40, so a little bit bigger, but not a lot. Another difference between prokaryotes and eukaryotes is that in eukaryotes, you have endoplasmic reticulum. You have rough and smooth, and a lot of ribosomes are embedded in the rough ER in eukaryotes. Prokaryotes don't have any endoplasmic reticulum, so all of their ribosomes are free floating around in the cytoplasm. In our prokaryotes, you can also have inclusion bodies, and what those are essentially are just little clumps of material inside the cell. They can be anything. They can be nutrients, they can be gas, they can be magnetic compounds, lots of different things. One thing they do is they help reduce the buildup of osmotic pressure by increasing your solute concentration inside a cell. So for example, in bacteria that live in very iron-rich environments, they will have usually iron inclusion bodies inside their cells. Here are a few examples. Another really unique thing that some bacteria, but not all, can do is make endospores. Endospores are essentially dormant, non-vegetative cells, meaning they're not growing, they're not metabolizing, they're not doing anything like that. They're essentially formed when environmental conditions are bad. There's not enough water, there's not enough nutrients, things like that. Essentially, the bacterial cell, when it encounters an adverse environment like that, it will create this very thick coat around its genome to preserve itself. And then it exists like that for a long period of time. Okay? They form a protective coat around their genome, and they essentially shed the rest of the cell. In this protective coat are also enzymes, ribosomes, pretty much everything the cell needs to survive, but it's in kind of this really hard shell as opposed to being in a normal cell. And that process is called sporulation. There's a video of it here for you that I'll put in the module as well. And then the reverse of that, when conditions are good, the endospore can shed its coat and then turn back into a normal functioning cell. All right, let's talk a little bit about the evolutionary transition from prokaryotes to eukaryotes. So how did eukaryotes first arise? The very first organisms on Earth that were cells were prokaryotes. And so from them, eukaryotes had to arise. And we think that that happened through a process called endosymbiosis. There are two really great videos here about endosymbiosis that kind of help the process make more sense. Essentially what endosymbiosis is it's a big cell taking up smaller cells that have specific functions, and those smaller cells that it's taken up actually become things like organelles inside the big cell. All right, so let's talk about eukaryotes. So what are they? They are cells of things like protists, algae, fungi, plants, and animals. Some of them are unicellular, some of them are multicellular. They always have a nucleus. That is the key. And they have other membrane-bound organelles. things like endoplasmic reticulum, Golgi, lysosomes, peroxisomes, mitochondria. All of those are unique to eukaryotes. Their genome, instead of being in one big circle, are multiple rod-shaped chromosomes. We call those linear chromosomes, and they're much, much larger than prokaryotic cells because there's so many more things inside them. Eukaryotic cells have many different shapes and sizes, even within species. This just highlights some of them. You can see all of the differences. Some of them have cell walls, but some of them don't. No animal cells have cell walls, right? Plant cells have cell walls. Algae have cell walls. Protists don't have true cell walls, but they have kind of a covering around the outside of them. Fungi have very strong cell walls. So it's really different from one category of organisms to another. There is no peptidoglycan in any cell walls that is unique to bacteria, which is one of the reasons that antibiotics won't target eukaryotic cells, because there's no peptidoglycan in eukaryotic cells. Most cell walls, if they have them, are made of carbohydrates, things like cellulose, chitin, glucan and mannan are different kinds of cell walls that yeast cells can have. Remember, yeast are single-celled fungi. And our protists, they don't really have true cell walls, but they do have this kind of stretchy support within the right underneath the plasma membrane inside the cell called a pellicle. Plasma membranes are really similar between prokaryotes and eukaryotes, except that plasma membranes in eukaryotes can contain sterols like cholesterol. They give it a little more stability. They make the membrane less fluid and more rigid, and they can help the cells, again, resist lysis, especially if you don't have a cell wall. That's an important thing to have. Crossing the membrane exists pretty much the same way in both species. A couple of cool things that eukaryotic cell membranes can do are endocytosis and phagocytosis. Endocytosis essentially is when you are bringing things into a cell. The membrane surrounds a particle and then it kind of engulfs it into the cell. In phagocytosis, that membrane is extended around things, like how an immune cell can pick up a bacteria from the environment. It extends the membrane out around a particle and then kind of grabs it and brings it in a little sphere called a vacuole. Eukaryotes can also undergo pinocytosis, where they make a little kind of a fold in their membrane and then they pinch that off. And those are much smaller than phagocytosis vacuoles. It's just kind of like taking small samples of the environment. And then the opposite of that is when they're secreting things from their cells. Those things to be secreted are packaged in vacuoles and they are brought to the plasma membrane, and then the vacuole fuses with the membrane and kind of spits out its contents into the extracellular environment. And here's a picture of that happening here. It's pretty cool. All right, so the hallmark of eukaryotes is the nucleus, right? The nucleus is where all of the cell's DNA is. It's surrounded by this two-membrane complex called the nuclear envelope. Inside the envelope, there is a mesh of filaments that give it stability and structure. That's called the nuclear lamina. You might have heard of that before. It's essentially two phospholipid bilayers, right, with a little space between them. And inside the nucleus is what's called the nucleolus. That's the dense region where ribosomal RNA synthesis occurs. Ribosomes have their own genetic material, and it's where ribosome assembly starts. So that is kind of, when you're looking at a nucleus, that's kind of the really dark part inside the nucleus. Eukaryotic ribosomes, speaking of ribosomes, are a little bit bigger. They function the same way. They make proteins, right? Some are in the cytosol. A lot of them are on the rough endoplasmic reticulum. And the ER starts making what's called the endomembrane system. This exists exclusively in eukaryotes. It's this big long system of tubules, sacs, and flattened discs. It's essentially from the nuclear envelope to the rough ER to the smooth ER to the Golgi. Okay, that's the endomembrane system. The endoplasmic reticulum is kind of all of these, we call them flattened sacs because that's really what they look like when you look at it. And in the ER, you have mRNA that gets synthesized and moved out of the nucleus. And in there, they can be turned into proteins. They can be turned into proteins and then transported to the Golgi. There's a few different functions the ER can have. The rough ER is the one with all of the ribosomes on it. It's connected to the nucleus and is really important for transporting proteins into and out of the nucleus. It also is where newly synthesized molecules and proteins are stored. The more proteins are needed by a cell, the more rough ER there is. Okay, smooth ER is for storage. There's no ribosomes there. It contains enzymes needed to make fats and steroids. So important, but not for protein synthesis. After the ER is the Golgi complex. Those are, again, more of these kind of flattened sacs, if you will. They can modify our proteins by adding things like carbohydrates and lipids to them, creating glycolipids and glycoproteins, and they can package their products into little vesicles to be secreted out of the cell. A couple of other really important membrane-bound organelles inside eukaryotes are lysosomes. Lysosomes are kind of a sphere of phospholipid bilayer, and inside are very, very powerful digestive enzymes. They break down molecules, they digest bacteria, they can fuse with vesicles of things that are taken up into the cell to digest whatever got taken in. This is one of the ways that phagocytic cells in the immune system can kill pathogens that it takes in. Has a very, very low pH inside the lysosome because a lot of digestive enzymes work better in acidic conditions. And then we have our peroxisomes. These are small membrane enclosed spheres that contain chemicals to help break down toxic oxygen species that arise when cells undergo metabolism. And then we have cytoskeleton, which are just really tiny little tubules and fibers that give the cell structure and support. A really important organelle unique to eukaryotes are mitochondria. Cells can have hundreds of mitochondria. If they require more energy, like a muscle cell, they're going to have a lot more mitochondria. Mitochondria are the powerhouse of the cell, right? Most people have heard that. It's where the ATP is made in a eukaryote. Prokaryotes don't have mitochondria. They make the ATP in their cytoplasm and across the plasma membrane, which we'll get to later. Mitochondria have two membranes. They have the outer membrane, and they have an inner membrane that's actually very large, but it's kind of crumpled up inside, gives it more surface area. That's kind of where the ATP is made. Kind of like mitochondria are chloroplasts. These only exist in photosynthetic cells, obviously. That is where the sugar or glucose is made from the sun, right? And then we have our extracellular matrix. So kind of like how bacteria can make glycolax, eukaryotic cells can secrete this kind of gooey, carbohydrate-rich substance as well, but we call it extracellular matrix. Some of the extracellular matrix can form things like basement membranes, or it can contain things like collagen or fibronectin. It allows cells to exist in tissue, so really, really important in animal cells. Eukaryotic flagella are a little bit more flexible than prokaryotic, and they move like a whip or a wave rather than just spinning in circles like a prokaryote does. Eukaryotic cells can also have cilia, which are tiny, tiny little tails, kind of like tiny flagella. They move very rapidly. They can either move particles away from cells or towards cells, You're probably most familiar with the cilia in maybe the airway that moves things up and out of the airway. Here's a really great summary slide of differences between prokaryotes and eukaryotes. A good thing to come back to when you're studying. Okay, they both have plasma membranes, DNA, ribosomes, and cytoplasm. But this is not drawn to scale. Prokaryotes are much, much smaller than eukaryotes. Eukaryotes are larger because they have so much stuff inside them. So in summary, where do cells come from? Cells come from pre-existing cells. They don't arise out of nothing, right? So cell theory, cells come from pre-existing cells rather than spontaneous generation. Okay? We know that eukaryotes evolved through a process called endosymbiosis, where they took up smaller cells, and those smaller cells became organelles like chloroplasts and mitochondria. Important parts of prokaryotes, their cell shapes and arrangements. Those are very unique to prokaryotes and will be very important for us. Also very important are gram-positive versus gram-negative cell walls. Cell walls, remember, help protect from osmotic lysis in hypertonic or hypotonic environments. And then that is the end. Thank you very much.